So let me introduce our speaker. We're delighted to have Dr. Peter Chang today. He received his MD from Northwestern University Feinberg School of Medicine. He completed a residency in diagnostic radiology at Columbia University in New York City at New York Presbyterian Hospital. And he did a fellowship in neuroradiology at the University of California at San Francisco as part of their T32 research program. Dr. Chang is currently an assistant professor in residence for the departments of radiological sciences and also for computer science at the University of California at Irvine. And he's the director for a new center for artificial intelligence in diagnostic medicine, which is a multi-specialty initiative to develop and integrate artificial intelligence technology across the UC Irvine healthcare system. Dr. Chang has won numerous awards, including RSNA Rankin Resident Research Award, the Roger Bowman Award for Best Scientific Paper at SIM. He's won that multiple times. The President's Award for the American Rankin Race Society, and he was the Mackay International BRATS Challenge winner. That's the Brain Tumor Segmentation Challenge, among other awards. He's also a co-founder of multiple AI startups, including most recently Avencia AI, which is a company focused on deep learning for medical imaging diagnosis. His unique perspective that you'll hear from him today arises from experience both as a radiologist physician, but also as a full stack software engineer with over a decade experience in building FDA cleared tools that have been used in hospitals around the world. He's a wonderful speaker, wonderful teacher, and today he's going to speak to us about deep learning in radiology, emerging trends, and new research. So Dr. Chang, please go ahead.
Thank you, Kathy, for that very generous introduction. Appreciate it. And I certainly appreciate the opportunity to be here with you all today. Oh, I think my screen share stopped. Try it again. All right, you guys can see my screen, hopefully. Yes. So anyways, when Kathy reached out, I had both the fortune and the task of coming up with a very broad topic, right? I was kind of given free liberty, and sometimes that's a blessing and a curse. That being said, I took that as an opportunity to kind of cobble together some of what I consider to be the emerging trends and newer areas of research in this field. I'm going to start out by just covering a little bit of some examples of tools we're using in the clinical workflow, both to highlight the type of work that we're doing and again, mostly set the stage because there is quite a bit of work that we need to do in this field, despite the progress we've been able to make in the past several years. So that'll be the overall trajectory of what we're covering today. These are my disclosures, as Kathy also had mentioned on a previous slide. So I'm going to, again, dive right in here with this first question, which is, what are some of the example tools that we are using here actively at the University of California, Irvine? Again, without going into an exhaustive list, I'm just going to highlight two categories just to give you a flavor, again, setting up the stage for what we'll talk about later. The first category of tools relates to the evaluation and characterization of COVID patients. This is in line with what I'm sure is the research interests now of many groups across the country. The very first tool here is a simple non-imaging based risk score calculator that you can sort of see here. This tool was designed to predict the likelihood, the eventual likelihood of a patient ever needing an ICU admission or a ventilator specifically. And that risk is derived from a number of clinical risk factors shown here, including the number of various comorbidities, several routine lab values, and then a handful of what our algorithm ultimately identified as COVID-specific labs. These are labs that in our retrospective initial analysis had very high correlation with the outcome of interest. While some of these are a bit obscure, I agree. After our initial work was presented to the hospital leadership, we actually created a separate order set for our clinicians so that with a single click of the button, all 5 of these labs could be ordered at once. Here's a screenshot of the publicly available tool, which you guys can also take a look at this link. You'll notice that this screenshot here is quite old, dating from, I believe, August of 2020. As you can see here, the tool has been around for a bit of time, and it has been used in the interim to screen every patient with COVID positivity in our emergency room to date. Similarly, we're also working on some COVID type tools for chest radiograph evaluation, more specifically the detection of pneumonia. I'll emphasize here, this is not specific to COVID, but just general pneumonia. The algorithm is overall fairly accurate and more importantly, highly specific for the detection of pneumonia. And while some of you out there might point out that pneumonia is often not a diagnostic dilemma, there are a few cases where the diagnosis can be quite subtle. Here's an example of what might to some be a completely normally aerated right lung, but in fact there is a pneumonia here in the right middle lobe that is a little bit more obvious now that you kind of know where to look. As you see, the algorithm can perform two things separately here. One is segmentation of the right and left lung fields, as demarcated by this green line here, as well as a heat map or a segmentation corresponding to the suspected area of pneumonia involvement. This allows us to calculate what we've called the lung injury score, so a percentage of the lungs estimated to be involved with infection as a ratio. Here's some general statistics here, but the general important thing that we're able to prove over time is that this degree of lung involvement, as estimated just based on that 2D radiograph, does end up correlating to a number of clinical outcomes shown here in these various graphs, including, for example, the likelihood of death. This is the blue line here. Or the likelihood of an ICU admission requiring intubation, right? So again, in general, as a lung injury score goes up, the risk of any of these clinical outcomes does trend upwards. The second quick category of tools I want to highlight are those used to help enable stroke triage. So I am a neuroradiologist by training, so this is an application and field of work very near and dear to my own heart. To provide a bit of clinical background of what I'm talking about here, any suspected patient with stroke that comes to the ER needs to have a couple of key questions answered in the following order. Those include the exclusion of acute hemorrhage or brain bleed based on a non-contrast head CT, followed by the detection of a very specific type of occlusion or clot in the proximal large vessels of the brain. And then finally, if both of these two check out, you want to evaluate the amount of infarcted tissue or the ischemic core, the amount of brain that has already died, with the idea that if too much brain has been involved already, the likelihood of benefit of some type of anticoagulation or reperfusion therapy may not be as high. And it turns out that each one of these steps are in fact things that deep learning can help solve quite well. Here's just a quick animation here showing you two of those applications, the identification of hemorrhage and a proximal large vessel occlusion. On a simulated work list here, you can see the speed at which such an algorithm is able to filter a very busy work list and sort of hone your attention on the positive cases of interest. If you scroll through both of the exams, you'll find that indeed, this patient on the left-hand side of the screen has a small amount of subarachnoid hemorrhage here at the left brain vertex, whereas this patient on the right-hand side of the screen has a proximal large vessel occlusion right here at the right M1 segment. These are actually screenshots of emails that the algorithm is able to send into our inbox. This was just taken within the past 1 to 2 weeks, showing you, again, some small hemorrhage and a positive large vessel occlusion. I'm not going to go too much into the accuracy, but suffice to say, both our team here in validating our own tool and many others have shown the reasonable accuracy, raw sensitivity, raw specificity type values of overall algorithm performance. This is again for intracranial hemorrhage at over 95% accuracy in a prospective test set here. And here's the same data for large vessel occlusion, very high sensitivity and specificity values in our prospective deployment type environment. So again, you can reference the study if you want to know more, but in general, these are in line with what a lot of groups in the country are describing. And then finally, this is our tool for aspect scoring. So again, this is the process by which you're trying to estimate the amount of infarcted or dead brain tissue. This algorithm specifically does this by looking at the amount of darkness or low attenuation within the brain, as well as the amount of mass effect or brain swelling that occurs at any given location. The final result is this. So this is the secondary capture that is sent and archived into our PAC system. You will see both a aspects template. So these are the different affected areas of the brain, an atlas of key anatomic landmarks, as well as a heat map corresponding to the area of suspected acute infarct or infarcted tissue. This allows us to calculate a very specific aspect ratio. So basically a score of how many regions of the brain are affected. And on the right-hand side here, you see some quantitative information about each of the different affected regions. Now, I will point out that in addition to the work our group has done, certainly the area of stroke imaging triage is very popular, and there are a number of commercial solutions out there, just a handful of which are listed here in this table. This is drawn from a review article written by our group. So if you're interested in sort of how these different commercial entities compare and what the similarities and differences are, certainly I recommend you taking a look at some of this information shown here. Now, at this point, I will also point out that one of the biggest drivers for adoption and demand for stroke imaging tools, like the ones you just saw, including at our own hospital, is this very first CMS reimbursement code that was focused and targeted for a deep learning application. The specific mechanism here is a temporary new technology add-on payment, which essentially encourages the use of new technologies for a 1 or 2 year period of time. This was announced late last year, September of 2020. And again, the application specifically that was targeted by this first NTAP approval is large vessel occlusion. So the detection of those clots in a very specific location in the brain. The NTAP is specifically for an amount of up to $1,040 beyond the standard MSDRG reimbursement. What that means is that the payment really only comes into play when a patient is admitted into the hospital, is treated for a stroke with a particular type of thrombectomy procedure, and the total amount that the hospital has accrued in charges exceeds what is typically reimbursed. In that case, the hospital may be eligible for up to $1,040 beyond what is traditionally paid. So again, not necessarily every suspected stroke patient that comes and is triaged by the tool, but nonetheless, even this modest amount of reimbursement was enough, again, to significantly drive the demand for this very specific use case in just the past year. With an eye towards the future and understanding that these types of reimbursement will likely contribute to the landscape and where algorithms succeed and fail in the future, it should be kept in mind that the NTAP here was granted for the very specific reason that this application has a demonstrated improvement in patient outcomes over the baseline standard of care. So it's not just clinical efficiency or report turnaround time, but in fact, a demonstrable outcome benefit to the patient, right? It's a very stringent threshold to meet, but certainly something that we should all think about as we're developing these new tools if we want to tap into some of this potential reimbursement pool. All right, so anyways, that was, again, a brief highlight of types of tools used in the clinic, again, at our center and probably at many others across the country. It seems like we're making great progress and there's nothing that could possibly go wrong. But in fact, most of our time is spent here in active research, trying to solve the many problems that we're just beginning to uncover as we start to use these tools clinically. So what are some of these problems? Well, I should emphasize here first that while I showed you two tools that we're using, or two categories of tools we're using clinically, the vast majority of tools, including the ones we build, have not been evaluated in a realistic clinical setting. And when they are, the problems that we see are quite varied. So here's a handful of key questions I'm going to address in the subsequent slides. But in general, these are things like the inability of an algorithm to recognize clinical context, or the inability of the algorithm to tackle edge cases or unusual findings that come up only when you test on a large consecutive cohort. There are issues, of course, related to the generalizability of your model to data outside of your own institution, model interpretability or explainability, as well as workflow integration. Now, I will also point out that the way in which we evaluate these tools is also an area of active investigation. In other words, while most of our own work focuses on either sort of standalone retrospective type of evaluation, or if we're lucky, a fully integrated prospective evaluation, again, which has mostly come on in the past maybe 12 to 18 months at our institution. It turns out that this third bullet point of continuous monitoring might be the most important and yet most challenging piece to solve. In other words, is there a way for our team to monitor for potential algorithm or model degradation over time, especially as different hardware manufacturers become installed in our hospital, or local clinical practices or guidelines change, right? Is the algorithm still sort of contributing and adhering to the standards that we initially set out when we first validated the tool? To highlight the difficulty of this problem, consider that the only way currently that we have to test if an algorithm is doing a good job is that we need to manually label a large number of cases and dedicate an individual or a team's time to carefully go through all that data at fixed intervals. This simply is probably not a feasible solution long-term, either when you have many different applications all running in parallel, or if you start moving into the sort of more common, smaller community practices where such manpower would simply not exist. So this is certainly a very, very active and interesting area of work that our group and many others are engaged in. Now, again, I want to highlight now some of those issues and challenges that I just described on the previous slide. The first one here is the inability of our models to truly understand what I consider clinical context. In other words, while our algorithms are very good at making findings, pointing out abnormalities or things that one should pay attention to on the screen, the ability of the algorithm to translate a finding into a formal diagnosis, right, which requires a lot of clinical synthesis of information, that is very challenging. So consider this example, a patient that walks into the ER with clinical signs and symptoms suggestive of acute stroke, and this results into axial CT slice of the brain. Now, to many of us out there, we might actually point out that this head CT looks completely normal. And the reality is that in an acute stroke patient, that may completely be the case. That's a completely compatible type of thing to see, a completely normal head CT, because we know that acute stroke patients may present with absolutely no finding. In fact, the purpose of a head CT in this case is to screen for other abnormalities. But the diagnosis in the example of this normal head CT is absolutely consistent with acute stroke. By contrast, here is a small amount of acute hemorrhage that has been successfully and accurately detected by a deep learning model. This amount of high density here within the posterior left temporal lobe. But this acute hemorrhage finding is in fact not an emergency. It turns out if you look elsewhere in the brain, in fact, there's a significant portion of the anterior temporal lobe that has been just removed by a neurosurgeon. And in fact, here's the part of the skull that the neurosurgeon entered in through, so a craniotomy defect. In this type of situation, when a patient has just come out of the operating room, nearly every patient will have a expected small amount of postoperative hemorrhage. And in a busy academic setting, much like ours, and I'm sure yours institution is in line with this, the majority of all our hemorrhage cases actually come from this type of example. They're not, a patient coming into the ER with a new diagnosis hemorrhage is actually in the minority. So here's another, again, classic example of where the finding itself, which in fact has been accurately identified, does not necessarily lead to a straightforward diagnosis. The second challenge that we're seeing here are issues with technical implementation in terms of at least sending the right studies or finding the right studies for my model to evaluate. Consider that in a development environment, in a very carefully clean and curated environment, one that we use to build models, most of our tools have the luxury of a data set that has been carefully identified and selected for the task. Now, despite this initial model, which may perform very well in this isolated setting, in a realistic setting, it might be difficult for you to actually identify the correct types of images to even send to your model in the first place. I don't know how many of you have worked with this before. I'll say that in our deployments, we've spent upwards of 3 to 6 months building a robust pipeline to be able to carefully filter exams as needed to properly send to our algorithm. These problems specifically here include things such as sequence identification. So if you have an MRI or a CT scan with multiple series, right, and your algorithm was trained on just one of those series, how do you go through and filter those effectively so that you're only left with these series or sequences of interest? And then a second quality control step here, which attempts to assess whether the amount of contrast that your model requires is in fact administered, whether the exam covers the relevant anatomy of interest for an algorithm to make a good diagnosis, or perhaps the amount of artifact on the image, right? These are things that radiologists do subconsciously at the workstation as we're evaluating studies, but not something that our models traditionally have been built to accommodate. So this is another significant source of error that we see when we start turning on tools in the clinical workflow. Another significant problem that many of us in the field here will recognize is the issue of generalizability. So while our models tend to perform very well on data sets or cohorts that are similar to that used for algorithm training, in general, the algorithms are extremely brittle to data from other institutions or that differ significantly in some way from that training set. Now, the challenges here are unfortunately that most of our tools, most of our applications that we are building today are in fact only built on these small single institutional data sets, and that the types of curation that are required to build these models are very tedious. They're inconsistent, which means there's usually an iterative process that needs to be applied to consolidate annotations from different viewers. And the data sets that we end up building at the end of all this effort are only relevant for a narrow use case, right? So all of these issues contribute to the relative limited ability of our models to generalize. Now, because of this, our group and a few others, quite frankly, in the country have started now to aggregate resources and try to come up with very large generic data sets across many different disease processes. This so includes over 3 million exams in our PACS database that we have a copy of in our research environment here at UC Irvine, as well as a very large number of exams spanning various entities that you see here. This repository of data that we're starting to acquire and archive for research purposes gives us a kind of a unique opportunity here, or perhaps a direction towards building what I would consider to be a radiology specific version of a ImageNet-like database. Now, ImageNet here, for those unfamiliar, is in reference to the very large labeled data set that many non-medical industry state-of-the-art algorithms are both trained and benchmarked against over time. The core data as part of the ImageNet database forms the basis for the ImageNet challenge used to evaluate many different applications annually. Now, the rationale here, the benefit of such a large and generic data set here is as follows. So you have, number one, an advantage of large multi-institutional data sets, all aggregated and centralized into one place for algorithm development. Furthermore, if you follow the model of ImageNet type algorithms in the real world, you'll know that by and large, most of the algorithms that you develop don't need in fact, to be trained on a large data set, because you are able to benefit from a large and powerful pre-trained algorithm that has drawn from the experience of your large database. And for your specific use case, all you need to do is fine-tune the algorithm for a specific endpoint. This type of approach, where a handful of groups develop essentially the heavy lifting type big models on the big data sets, and then disseminate those pre-trained algorithms for fine-tuning to the vast majority of researchers. That model is, again, the standard in fields outside of medicine. And I think if we want models that are able to adapt and generalize the way many non-medical algorithms do, I think this type of ImageNet radiology database may be very valuable. And at this point, I'll say that, you know, while our group has certainly some effort and resources devoted to a project like this, such a project, in order to be successful, will really require the collaboration of many folks and experts around the world. So certainly something to think about for those of you out there who may potentially want to team up and work on something like that. Now, as an aside, there will be several of you out there who might question why we're reinventing a ImageNet type database for medical imaging, when in fact there are very good databases like this that are non-medically based. And so with two quick slides, I want to highlight some of the issues that we see when we try to apply an ImageNet algorithm to our radiology imaging. This is a diagram of the GoogleNet or Inception architecture, one of the very earlier versions, I should say, of the Inception architecture. I'm just using this as a demonstration because many ImageNet algorithms do have a similar type of topology. In these type of algorithms, you'll notice that as we know, the small or high frequency type features, these are the things that are learned early on in your model, things that associate with your borders and edges, and again, these low level, high frequency features. Whereas deeper in the model, after many subsampling operations, right, these features are by and large and low frequency type patterns, which quite frankly are the dominant types of things you see in real world data. So if I wanted to detect the presence of a dog in these two example photographs, that evaluation, that characterization, again, is typically the sum of a macroscopic or low frequency type pattern across the whole image. Whereas in our world, more often than not, we're interested in very subtle, small findings that are only present on a very focused area of the image. So these high frequency features, in fact, predominate. You'll notice that in this model, not only is there a lack of modeling capacity early in the model, in fact, many of these models will aggressively downsample in the very first one or two layers, right? In essence, losing almost all of that high frequency important information that we need for many of our different diagnoses. Additionally, imaging data out in sort of the real world oftentimes involves what I consider a single step or perceptive type interpretation. In other words, you look at the image once and you sort of synthesize a number of global features to make a very quick reflexive prediction. Radiology imaging, by contrast, often requires a more interpretive or multi-step type of pattern detection. In other words, if we were to look and assess for the satisfactory position of an endotracheal tube, in fact, what we will need to first do is look and identify the presence of an endotracheal tube. We'll also need to localize and identify relevant landmark anatomy based on our knowledge of where these tubes should live. And then we typically, after all of that, we'll need to make a third assessment of the correct distances between these two landmarks, either manually by making a measurement or at least looking separately at that distance to see if it falls within a normal range. This type of interpretation, again, is in my mind, very different and limited in terms of that generalizability when you look at non-medical types of data. All right, well, so the next part, I'm gonna now go kind of backwards, because I just spent a few slides talking about the importance of centralizing large data sets and the potential advantages they have. But I will admit that at the same time, this type of model has significant bottlenecks. So data sharing in our world has oftentimes been difficult due to various cultural, technical issues, or even simple baseline problems with de-identification, right? And so because of this, the idea of a centralized large ImageNet radiology database may in some ways be challenging. And so in parallel, there are other creative multi-site training type paradigms that I want to talk about. The first, of course, is very simple algorithm fine-tuning. In this type of paradigm, a model which is trained first on a very large database, here I'll mark as database A, is essentially trained to near convergence, followed by a focused fine-tuning effort on your own specific institutional data set labeled here as B. This is, again, a fairly standard, common approach that exists in many non-medical contexts and is probably the easiest to implement out of the different techniques we're about to describe. In addition, there are several related distributed or federated learning techniques that I want to introduce to you next. Federated learning and distributed learning, again, two very similar types of ideas here, are as follows. So with federated learning, a single model learns asynchronously, so independently from one another, at multiple different sites. So this single model, for example, here, can either be sent in serial to institution A for some edits, then institutional B for some additional edits, and then finally to institutional C for yet a third round of edits. That's one type of implementation. A second option here is for a model to actually be sent to all three institutions in parallel. All three institutions will perform some training for a predetermined amount of time. And then after the independent phase of training, all the updates get aggregated, get sent back to the central model for some sort of clever way of taking the average or updating the model using all that different data. Distributed machine learning is a very, again, complementary and similar type of technique, where the difference now is that the single algorithm learns synchronously or at the same time at all three institutions. So a single model never really has to leave. A single model gets to live in the cloud. And what happens here is that simultaneously in real time, each iteration step calculated at each individual institution gets sent to a central model. And again, this one model is learning as if all three data sets are in the same location. I'll point out here that while distributed machine learning leads to overall a little bit more stable training, as you might imagine, you'll need a very high-speed interconnect between all these institutions to be able to support real-time distributed training. Whereas a federated machine learning technique is a little bit more feasible. You're able to train with reasonable accuracy using this approach, using standard types of hardware and network connectivity. Fortunately, our site, and in fact, all the UC sites are on a a special Pacific Research Platform network spanning basically 10 to 40 gig wire at all campuses, allowing us to implement that distributed machine learning technique that I just described, where one machine essentially acts in synchrony with all other campuses at the same time. Our results here that we've seen are actually very favorable. The results are very stable, and they scale quite well to multi-GPU, multi-institutional type of training sessions. Related to this is one additional paradigm or concept I want to throw out there, which is the idea of continuous learning. In this type of model, consider a generic algorithm shown here at time 0, which has been already pre-trained and converged on some data set. Subsequently, the model gets deployed hypothetically at 3 different hospitals. And upon deployment, the model takes some of that local institutional data and does an incremental fine-tuning or update. Right? At this point, the 3 models now, because of exposure to different data sources, are incrementally different than one another. Over time, this model, as you might expect, may continue to update and continue to diverge from the different models at the various institutions. What this means necessarily is that this model from Hospital A may not generalize well to the other models, but for its own specific institutional data, that model has actually successfully overfitted, if you will. Now, this approach might seem exotic, but consider that a precedent can be seen if you look at the way our hardware manufacturers, our CTs and MRIs, are actually implemented in each of our hospitals. In other words, while many of us share the exact same hardware and similar types of machines and possible configurations, each one of our hospitals then goes and takes that generic hardware and modifies it with institutional specific protocols or acquisition parameters, such that, again, the customizations are very carefully made to meet the needs of our specific institutions. The limitation, of course, here, and in fact, the limitation I've highlighted many times at this point, is that this approach would require manual annotations, right? Our standard supervised learning approaches will always require annotations to perform this type of fine-tuning, and this is a significant limitation. So at this point, I want to dive in and transition to a topic that, again, is a very active interest of my own, and that's unsupervised learning. So ways for machine learning and specifically deep learning models to learn inherent patterns to data, patterns that don't require a human to go out and point at a diagnosis or point at a finding. In our world, for radiology imaging, these inherent patterns tend to be things like anatomy or appearance of various tissues on T1 or T2 weighted images, for example, which are very highly consistent from patient to patient. One might imagine that an algorithm that has had the flexibility and freedom to look at millions and millions of patient exams might start to understand that these patterns are very consistent, right? And are reasonable things to learn. I won't go into a lot of the technical details here, but there are various implementation strategies for this. 2 very popular strategies are known as self-supervised learning and deep clustering. In self-supervised learning, an algorithm is simply attempting to, again, reproduce some sort of pattern that is inherent to the raw data itself. In this example, one might imagine an algorithm which is presented with sort of raw images with portions occluded, so a big chunk of the brain here covered with a black box. This self-supervised learning algorithm is then asked to draw in or repaint the missing portions of the brain here, with an understanding that satisfactory completion of this task necessarily requires that the algorithm understands what the missing part should look like based on the contextual surroundings, right? So it understands essentially that this is a slice that's at the level of the lateral ventricles with particular blood vessels that are known to exist in this area of the brain, and so on and so forth. I will point out that if you look at trends, again, outside of medical imaging, self-supervised techniques are among the highest performing models of really any type of deep learning algorithm, in my opinion, in the world. This particular model, the GP23 model, is a language model trained using a self-supervised approach on essentially all of the written text available on the internet. Again, I won't go into the details here, but suffice it to say that this language model can do some pretty remarkable things, including writing an entire article here without any human guidance. So it was simply presented with a single prompt of whether or not AI would eventually take over the world. Again, the output is a long prose essay published now here in The Guardian, which I recommend you taking a look at if you haven't seen before. You know, I will point out one observation, which is that you actually don't realize, if you don't know it, you won't realize this algorithm was written by a deep learning system until probably about halfway or 2/3 of the way through. So again, really quite remarkable. I'm just going to highlight a little bit of some of our own unsupervised work we're doing in our lab. Here's one unsupervised model for segmentation using a deep clustering approach, something I referenced on that previous slide. In other words, what we asked the model to do was look at a large number of abdominal CT exams and simply subdivide the abdomen into various compartments, right? We sort of let the algorithm do whatever it likes. This is the output here in C, and again, it doesn't conform to any anatomy that we know as humans, but it gets pretty close, right? In fact, if you look at the embedding of this organization and you look at things that we as humans know as anatomy, in fact, our anatomy does tend to superimpose and separate in this low dimensional space very nicely. Such that if you take that pre-trained model and show it one example of, again, the anatomy that we as radiologists know and describe, again, with just one exam, the supervised fine-tuned model here is able to perform whole body segmentation with remarkable accuracy. Here's an unsupervised paradigm where the model is asked to do two things. It's asked to generate what we call as a template space, so a generic template reference space. And number two, the algorithm is asked to perform a nonlinear co-registration to that discovered template space, as you see here. Now, because we don't know the template space ahead of time, because the template space is something the algorithm gets to decide on its own, I can't train it using a very standard supervised learning model where you just map your input to the template space, as nice as that would be. However, we do know that even if the template space itself is yet to be determined, the mutual information between the original space and the template space must be high, no matter what template space the algorithm decides to eventually use. So we can actually use this as a clever reformulation of the problem to, again, maximize the mutual information between these two potential spaces. And again, the algorithm using this approach is able to solve both of those tasks. Now, again, I'm going to bypass the math here and just show you some pictures. Here's an example where we tried using that algorithm to identify the pancreas, which is a very difficult organ to segment, as many of you might have experience with doing. Here would be two representative patients in the top and bottom rows here. The unsupervised model, after convergence, is able to take these two patients, map each voxel in these two patients to its corresponding location in a standardized template space, which maps approximately to something like this if you were to zoom out. And based on this, we're able to then reverse those mappings and go back and find the pancreas. Here's some heat maps showing the approximate location of the pancreas in two patients. And in our initial data here now, spanning several thousand patients, we're actually able to show fairly good and robust results using this technique. Now, with just my last few slides here, I want to highlight one final problem, which relates to interpretability or the explainability of your models. It turns out that in general, while it's very easy for you as a human to agree with a model. So a model says, I see a hemorrhage, and you scroll through and you see a hemorrhage. That's a fairly straightforward interaction. And we've seen in many of our studies that the radiologist is indeed faster in those cases. The problem arises when there is a discrepancy between the model and what the human sees, right? Invariably in those cases, the ability for the human to visualize what the model was looking at is critical. We've actually shown with good evidence that when you don't have that, the human radiologist spends more time looking at the image because we're trying to find the disease process or the entity that is not there. But we simply don't want to miss it because the algorithm thought it saw something, and we don't want to be the human that missed something the AI saw. So interpretability in some very specific contexts is absolutely critical. Now, from a practical regulatory perspective, it turns out that the most popular FDA clearance pathway, this computer-aided triage pathway, which is leveraged by nearly all deep learning startups today, this category does not allow for explicit image annotation. There are other categories, the more standard CADI or CADX, that we're familiar with that allow this, but this special category leveraged by deep learning companies do not. And we've seen that this is, again, a very significant limitation to the way that our radiologists ultimately interact with the algorithm. In addition, I will posit that as the algorithm developer, when I build a model that is able to show its work and to be very explicit about where findings exist, right? Those models actually end up having different types of errors, right? So two different types of errors I'd like to point out here. The first is what I consider to be a human-like error. This is an error where the algorithm technically made a mistake, but the mistake or the error in finding is equivocal. So if you asked a panel of 10 or 20 radiologists, it's likely that several of those radiologists might actually end up agreeing with the AI system. By contrast, there are what I call random errors. And these are things some of us, especially those making algorithms, will know. There are occasionally mistakes that your algorithm uncovers, which are completely consensus mistakes. So to even the most modestly trained physician, in fact, the mistake is quite obvious. Now, in addition, again, to the obvious legal liability that these two types of errors hold, I will again go back to my initial point, which is that algorithms designed to make an explicit representation of why a particular prediction was made, these type of algorithms tend to make very human-like errors. When it highlights a false positive hemorrhage, is usually just another bright thing in the brain. That's a distractor. Whereas algorithms that are designed to be more hands-off, to provide a global diagnosis on a slice level or on an exam level, those global type of algorithms can literally use any feature under the sun in any part of the image to make a diagnosis. And those type of algorithms do tend, unfortunately, to make the rare but nonetheless prevalent or real random type errors here. All right, well, I think that basically summarizes what I have today. I want to just close here with a slide here about the center that I direct and sort of our team here at UCI. This is a new center for AI in medicine built about 3 years ago here at UCI that I was essentially recruited here to develop. And our vision essentially is to act as a central team of both physical as well as human resources and various expertise to help clinicians and non-experts translate on their problems. It turns out that when you set out to perform a deep learning experiment, you need to solve a ton of little things. Some of these are very easy, but some of these are quite challenging. And quite frankly, you don't want to be solving these problems more than once in a single institution. You'll notice by contrast in pink here, the number of things that need to be redesigned or customized for a specific project are fairly limited. So with this in mind, our center tries to again, host and solve as many of these services as possible. Again, allowing our clinical experts and others with ideas and problems come to us, and essentially work together to build and solve various problems. So anyways, that's just a brief overview of what we're doing. This is my contact information if you'd like to reach out and chat and or perhaps collaborate on some of these research trends and topics I presented. We're always more than happy and very interested in working together. So please don't hesitate to reach out. I thank you again for your attention. I believe we have a couple minutes here for questions.
Well, Peter, that was fantastic. Real tour de force there, covering everything. It's fascinating that you're doing a lot of prospective testing, and that you had to work with vendors to get tools implemented into your current clinical systems?
Yes, in fact, we absolutely have. I tried to avoid any references to commercial entities during this talk, but absolutely, we're working. So we're working with two types of companies. One type of company would be sort of the more sort of what we call the generic platform or marketplace entities. We're working with a lot of those folks to be the sort of intermediate third-party entity that performs the integrations, but we provide the tools. So those are folks like Arteris here in the UC system. Many of us have that Nuance marketplace as well. And we're going live with Visage in just a few months here. Visage has some great options for deployment there. We are, of course, also working directly with a number of commercial entities which have tools themselves. I myself am co-founder of a startup company, and I have a lot of colleagues across the US and actually in Europe and Asia who have tools that we're very happy to test out and use and compare and really just get a feel of what's useful. The performances, as we've seen, can be highly variable depending on the use case, depending on where things were trained. And we've seen that there's really not a good way to understand the actual impact on your workflow except to try and use those tools. So we are absolutely engaged in many of those types of discussions.
Fantastic. And I was fascinated by your discussion around, a lot of folks are using sort of generic models, architectures, and starting with transfer learning, something that's been trained on cats and dogs and cars and whatnot. And, I hadn't seen a paper that discussed whether we expect models to perform better if they are trained innately on medical images, which you seem to suggest, which I think is fascinating. And the other piece, and maybe you can comment on that, and the other part of that question is, the FDA is very conservative about approving continuous quote unquote learning, but you kind of make the case therefore, well, give folks a model, let them work it and tune it to their site, and that's great, and that's fine. So could you comment about those two pieces?
Yeah, two very, very good questions. To your first point, it's actually a great blog article, I think, by the Google Brain team. And they compared basically all sorts of ways people are building medical AI models. And in general, they saw that transfer learning from ImageNet compared to de novo building an algorithm on a small focused medical data set tended to be very similar. Sometimes one did a little bit better, sometimes the other did a little bit better. Again, it kind of depended on how good ImageNet transferred in those cases. But what many people in the field recognize, and the third category essentially that's not tested, is pre-trained models from a very large dedicated radiology database. I would make the argument that that is absolutely critical. Most of the generalization issues that we're seeing now come from the fact, not that we don't have a lot of cases of hemorrhage or a lot of cases of stroke, but the fact that the model has not learned an innate representation of medical data. They don't understand what an artifact looks like, which is common across all modalities. They don't understand what beam hardening is or why certain areas around the bone just become bright. These are things that, in my opinion, could be solved quite well using a more generic pre-trained approach. And then again, you're going to then apply your more standard fine-tuning strategies after that to build that generalizable model. With your question for fine-tuning, continuous learning rather, I do think that's a big challenge. Fortunately, the FDA as part of a white paper released, I believe it was a year or a year and a half ago at this point, they've at least acknowledged the need to address continuously learning models. They haven't formally approved anything yet, of course, but I think that in my mind is probably one of the key areas that we'll need to see for these truly next generation type models. Right, what we're seeing now are fairly simple, one use case models, very narrow. We're getting modest results kind of as is, but if you wanted to be more ambitious, right, if you needed a model to differentiate between normal and abnormal exams across any potential pathologic finding across every vendor and possible implementation scenario in the world, I don't think it's possible to build an algorithm that does that very well, at least using our current technology today. And so I think this continuous learning approach is very valuable. I think injecting some unsupervised components of that continuous learning in order to make that actually happen in a realistic manner is a part of that picture. But yeah, certainly many things are yet to be solved too. So we'll see kind of where the field takes us in the next few years.
Fantastic. And it also speaks to, I think, the value and I think the requirement to have clinical people involved in this whole process. So we need to have more Peter Changs involved in our process. We have one question here from someone in the audience. Can you please talk a little bit about the usage and requirement of explainable AI in the FDA approved products?
Yeah, so the point I was making was interestingly almost the opposite. So rather than requiring models be explainable, the current computer-aided triage category, which is again happens to be very easy to obtain clearance for, and thus the default pathway for most startup companies, that pathway, in order to help have a quick turnaround for approval, limited validation type of data is needed, but the caveat is, again, you cannot annotate. You can't tell the reviewer or the human why a particular abnormality is the way it is. There is a slight wrinkle right now, which is that some companies have found a way that they can basically load the correct slice that the abnormality is found on. So when you click the study, it just happens to default to the slice with the abnormality without pointing to anything. So that's again a tricky workaround. But by and large, the language of that is that you can't mark things up, because once you mark things up, you're in the CATI or the CATX category, which requires much more stringent requirements. So that's currently, unfortunately, the state of what it is. If you look in Europe and Asia, that's not the case. And all of the tools that we make commercially, we have a US version and a European version, where the US version, we must turn off all of our annotations. So anyways, it's kind of frustrating. I think it's just a little bit of a lack of perhaps careful understanding of what the implications of explainability are. The one other comment I'll make here, because explainability is a very charged topic and people have all sorts of sort of preconceived notions. One is that, does it really matter that your model makes an explainable prediction if the prediction is correct, right? I would argue that for things that humans are already very good at doing, like looking for hemorrhage or looking for stroke, you're going to need to explain your work, because a radiologist ultimately is going to be double checking that prediction and agreeing or disagreeing with what you're doing. When you're moving to these superhuman tasks like predicting the likelihood of bad outcome in cancer, right, or things that humans just aren't very good at, it's likely that the features used to make that prediction are not explainable. There's some high level thing that humans don't really appreciate and understand well now. And I would argue that at some point that need for explainability will become less stringent. But again, today, currently in 2021, I would argue that it's a critical part to building that trust and building a realistic workflow that helps the radiologist.
Fantastic. And of course, the next realm is really tying all this to outcomes so that we can get reimbursement through that NTAP. I'm afraid we've come to the end of the session. I could ask you questions all day. This was a wonderful, wonderful presentation. And we thank you, Dr. Chang, very much for participating.
Absolutely. Thanks again for inviting me. It's a pleasure to be here.